Come on. Okay, sorry for being a little late. Are there any questions? No questions? Let's go ahead and begin. I'm sharing my screen now. So if you look at the schedule for May 13th, we are starting Chapter 8, Microbial Genetics. There is no lab today, but I will be there from 6:30 to 6:45 to answer questions. If you have any questions, make sure you log in between 6:30 and 6:45, because after that I'll be logging off. Let me remind you that Lab 8 is due this week, and also the infectious disease paper is due by 11:59 p.m. Saturday, May 15th. And today I should be sending you, during the lab, I should be sending you your first results from the first tests on your unknown project. Any questions about any of that? All right. I guess I will shut that down. So, can I get confirmation that you guys are seeing my screen?
Yes, we can see your screen.
All right, thank you. Oh, I don't have this in, sorry. So we're starting Chapter 8, Microbial Genetics. And as always, I give you a starting slide where I give you the major goals for the lesson and the rough lesson outline. Now, this is not all of the goals, all of the objectives. For all of the objectives, you should read the study guide for Chapter 8, and at the top of that section, I've got the objectives. Any question about that? Let me see that this is working right. Ah, it's not working right. I don't have the screen on. Come on. Just a minute here. I'm having a hard time. There we go. Now let me share the screen again. So know the terms for this chapter: a genome, a gene, genotype, phenotype, vertical and horizontal gene transfer. What's an operon? What's a promoter? An operator? Know what a structural gene is, what crossing over is, what transformation, conjugation, and transduction are. Be able to describe DNA replication, RNA transcription, and protein translation. And then lastly, be able to understand the different types of mutation. You'll do that, the different types of mutation, in a worksheet for Chapter 8. So we're going to talk about the structure and the function of the genetic material. We'll then move on to the flow of genetic information. We'll talk about how DNA replicates, and then how RNA transcribes. And then the next topic will be how a protein is translated. We will then move on to the regulation of a bacterial gene expression, how the genes are regulated, and we'll talk about operons in that process. And then we will talk about mutations, but that's actually something you will be doing in the worksheet. And then we'll talk about genetic transfer and recombination, and we'll end on talking about genes and evolution. Any questions about what we're doing? Okay, so genetics is the study of what genes are, how they carry information, and how that information is expressed, and how genes are replicated. A gene is a segment of DNA that encodes a functional product. Not all of the genetic information is a gene. Okay? Like this region here does not code for anything, at least not a functional product, which is usually a protein, but it can be RNA. Any question about any of that? The genome is all the genetic material in a cell. The study of genomes is called genomics. The genotype is the genes of an organism. For example, individuals with blue eyes, which is the phenotype, the expression of the genes, their genotype is lowercase b, lowercase b. That's their genotype. Individuals with brown eyes, which is the other human phenotype for eye color, bluish or brownish, their genotype is either capital B and lowercase b, or two capital B's. Okay? Any question about that? Obviously, the lowercase b stands for blue eyes, but it's recessive, so you need two. And then lowercase b is recessive to uppercase b for brown eyes. So an individual who's uppercase b, lowercase b will have brown eyes. And let me think about that. Both my brother and my sister-in-law were uppercase b, lowercase b, because two of their children had blue eyes. Any question about genotype or phenotype? Here we're looking at a chromosome. A chromosome is where most of the DNA is, all the DNA that's not in a plasmid is in a chromosome. And the chromosome is both DNA and its associated proteins. So most of the genes are on the chromosomes. Most bacteria have one chromosome. As you know, humans have 46 chromosomes. DNA is a polymer of nucleotides, meaning DNA is a whole bunch of nucleotides. And DNA has 4 nucleotides: adenine, thymine, cytosine, and guanine, or you can call them A, T, C, and G. I don't care how you call them. You should memorize them, but you could use either system. DNA is a double helix, meaning one strand of DNA is running this way, and the other strand of DNA is running that way. So they're in, what is that called? Anti-parallel sequence. And all it really means is that one strand is facing that way, one strand is facing that way, and you can say the 3' end is on this side, the 5' end on that side, the 3' end on this side, the 5' end on that side. 3 and 5' are the carbon in the sugar. So this would be the 3' carbon, and that would be the 5' carbon. When we look at the DNA, it's made of 2 molecules of DNA, or how do I say that? 2 sequences. One sequence is held together by the sugar phosphate backbone, meaning it goes sugar, phosphate, sugar, phosphate, sugar, holding the DNA together in one strand. The nucleotide bases are on the steps of the strand, and they don't hold the polymer of nucleotides together. What the nucleotide bases are doing is they are hydrogen bonding to the complementary nucleotide base on the other strand. So the 2 strands are held together by hydrogen bonds, and the hydrogen bonds happen between A and T, or you could say T and A, and C and G. Any question about any of that? And you should know that A and T have 2 hydrogen bonds, and C and G have 3 hydrogen bonds. Now, each hydrogen bond is not very strong and can break, but the DNA is held together by numerous bonds. So when this one breaks, these bonds, as well as that one, can hold the 2 strands together, and then this one can reform, and then that one can break. Any question about any of that? If not, let's move on to the vertical flow of information within cells. We'll talk a little bit more about the flow of information from DNA. The central dogma of biology is the information is stored in DNA, and then that information is transcribed into RNA, and then that information is translated into the information of protein. So the information flows from DNA to RNA to protein. And this is known as the central dogma in biology. Each of these molecules stores the information in their own language. We'll talk more about that in just a minute. Let's talk a little bit more about the flow of genetic information. As I stated, information can flow within a cell from DNA to messenger RNA to protein. That's how information flows within a cell. Information can flow between generations of cells, like when the information is flowing from parent to offspring, or parent cell to daughter cells. The information in the DNA of the parent cell is copied or replicated, and then the DNA moves towards opposite ends of the cell. The cell divides into 2 daughter cells, each getting a copy of the information in the DNA from the parent cell. So that is how genetic information flows between generations of cells. Now with humans, the information flows from 2 parents generally to 1 offspring, and that's because of sexual reproduction. Any question about any of that? Okay. DNA can also flow between cells of the same generation. And I've broken the picture in the chart to make it a little clearer. The DNA comes out of the parent cell, probably when this parent cell dies. And then the DNA is naked DNA in the environment. And that naked DNA can be taken up by a recombinant cell, which gains the DNA from another cell. And then if that DNA is expressed, we say we have a recombinant cell. And that is how the flow of genetic information can happen between cells of the same generation. Any question about any of that? So the information can flow within cells, between generation of cells, and between cells of the same generation. Now we talked about the flow of information within a cell. This is also called the vertical flow of information because the information moves vertically from DNA to RNA to protein. When the information is stored as DNA, that information is stored in nucleotides, and there are 4 letters to the alphabet, or 4 different nucleotides. And we actually mentioned that it's A, G, T, and C. The information then flows to RNA, and once again the information is stored in nucleotides, and there are 4 of them, but they're not the same as DNA. 3 of them are the same except that it's an RNA nucleotide instead of a DNA nucleotide. But one of them changes. We don't have thymine or T. It's replaced by uracil or U. And then the information flows from RNA to protein, and the information is stored in the amino acids. And humans have 20 different amino acids to store that information. And of course, the English language has 26 letters in it to store the information of the English language. And that's just to give you a comparison of how these languages are being expressed compared to English. So when the information is stored in DNA, the information would be something like, that is a dog. And then the information is transcribed into RNA, and the information would be very similar, but with a different dialogue. Something like, that is a dog. Hopefully you could distinguish the difference there. And then when the information is translated into protein, the information is changed into a completely different language. And it would be something like, quello è un cane. Any question about any of that? So we've talked about how the information can flow within a cell. Let's talk about how the information can flow between generations of cells. This involves the parent DNA molecule being replicated and then dividing into daughter cells. Any question about any of that? So DNA replication is said to be semi-conservative. What do we mean by that? It means we use one strand of DNA as a template for making the new strand. So the DNA polymerase binds to this strand of DNA, and the 2 strands separate. So this becomes a single strand. And then the DNA reads the nucleotide here and says it's a G, And so it brings in a C and then adds that to the growing strand of DNA. And then it reads the next nucleotide, which is a C, and adds the nucleotide G to the growing strand of DNA. And that's how this new strand of DNA gets made. And that is, the other strand is used as a template for building this strand. And that is semi-conservative replication. Semi-conservative replication means that if we have the original strand with dark strands of DNA, and the new strand is a light or white strand of DNA, the newly replicated DNA will have one dark strand of the original DNA, and then one white strand of the newly replicated DNA. And that'll be true for the other strand of DNA. And that's semi-conservative replication of DNA. That differs from conservative replication, which would be where in the daughter cell, one cell would have two of the original dark strands of DNA, and the other daughter strand would have two white strands of the newly replicated DNA. Conservative DNA replication doesn't occur. You only need to know about semi-conservative replication. And don't ask me what liberal replication of DNA is. I've never seen a model for that. Okay? Any question about any of that? So DNA replication happens when the DNA polymerase sees the original strand of DNA and sees the nucleotide base. This is an A, and so it'll put in the complementary base, which is a T, on the new strand of DNA which is growing. So the DNA polymerase uses the original strand as a template to make the new strand of DNA. Any question about any of that? And how that's done is a triphosphonucleotide is added, and the energy for attaching that happens with two of the phosphates coming off, and then the third phosphate attaches this nucleotide to the previous nucleotide. All right, so let's watch a little video of DNA replication. What happens is the DNA polymerase binds to the DNA, and the helicase binds to the DNA, and that separates the two strands, not helicase, the, what is that called? I'm drawing a blank here. All right, ligase, DNA polymerase, and the helicase, I think is what it's called. The helicase binds to the double-stranded DNA and makes it single-stranded, opens it up, and then the DNA polymerase can bind to the single-strand DNA and add nucleotides to the new strand of DNA which is being replicated. So watch the little video here, and this will probably be too loud for me, so I'm going to remove that.
DNA is a molecule made up of two strands.
Let me turn that up. That's awfully quiet.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Okay, any questions about any of that? I think I'll move that just in case we come back to it. But I think we got pictures here. So let's go through that step by step. DNA is copied by DNA polymerase. The new DNA is made in the 5' to 3' direction. So the DNA polymerase only moves in the 5' to 3' direction. Now that is okay for the leading strand because it is running in the 5' to 3' direction. And the leading strand is synthesized continuously. So except where the DNA polymerase begins and maybe where it ends, the DNA will be made in one step, and there will only be a gap, not a gap, but the DNA won't be held together where it is made, first made, and then maybe where it's first ending. Everybody see that? Now, on the other strand, it's running in the wrong direction, for DNA polymerase. And so it makes or copies that strand in short little pieces called Okazaki fragments, after the doctor who first discovered this. And wherever you start, as well as wherever you end, the DNA will not be held together in a covalent bond. And the important part I'm trying to say is that the ligase, which at the very end links the DNA together, will do that. It'll do it on the leading strand where it started, so that the old strand of DNA is linked to the new strand of DNA, or how do you say that? Close enough. And then where it ends. So the ligase on the leading strand may only need to work 2 times, but on the lagging strand, the ligase has to do it for each Okazaki fragment at the start and the end. So for each fragment, the ligase is working 2 times. And so the ligase is going to be working a number of times on the lagging strand. And it's called the lagging strand because the DNA is made in short little chunks, as well as the fact that the leading strand, the DNA can be replicated faster, so this strand leads the lagging strand, meaning this strand the DNA is replicated a little slower on this strand. All right, the process of making DNA is started with an RNA primer, and then the DNA is put down. That RNA primer will be removed, and the Okazaki fragments joined together by DNA polymerase and DNA ligase. A ligase links the DNA together with a covalent bond. Any question about DNA replication? If not, let's move on to transcription. Transcription is the synthesis of RNA from DNA. Transcription only occurs at the genes. So all of the DNA, which is not a gene, will not be transcribed. Now, in prokaryotes, most of the DNA is genetic information. I should say, at least of bacteria, most of the DNA does code for something. So most of the DNA is genetic information. So what I'm trying to say is most of the DNA will be transcribed. But in a eukaryote, most of the DNA does not code for anything. So most of the DNA in a eukaryote will not be transcribed because only the genes are transcribed. And that's very different from DNA replication, where all of the DNA is replicated. But in transcription, only the DNA, which codes for a gene, will be transcribed. So transcription is the synthesis of RNA from DNA, and DNA can be transcribed to make messenger RNA, or mRNA, or tRNA, or transfer RNA, or ribosomal RNA, or rRNA. How does the RNA polymerase know which RNA to make? How does the RNA polymerase, reading the DNA know to make messenger RNA, or tRNA, or rRNA? Anybody have a foggy guess?
I mean, I can take kind of a shot in the dark.
Go ahead.
So either it's in the DNA or maybe, yeah, I'm not really sure. Maybe it must be in the DNA, right?
Yeah, you are correct. It is in the DNA. If the gene codes for a protein, then that gene will be transcribed as messenger RNA. But if the gene codes for tRNA, then the gene will be transcribed as tRNA. And if the gene codes for rRNA, it will be transcribed as rRNA. So it is in the DNA. It's whatever the gene is coding for. Will depend on what RNA molecule is made. Everybody see that? Well, I'm hoping everybody sees that. So transcription begins when the RNA polymerase binds to the DNA. And RNA polymerase binds to DNA only at the promoter. The promoter is a region of DNA that codes for the RNA polymerase to bind to it. And then transcription proceeds in the 5' to 3' direction, meaning RNA is made in the 5' to 3' direction. Now, eukaryotes have 2 strands of DNA, right? Usually only one of those strands is read to make RNA. And the RNA will be made in the 5' to 3' direction. Any question about any of that? The RNA polymerase continues transcribing RNA from the DNA of a gene until the RNA polymerase reaches the terminator. The terminator is the last part of a gene. And what the terminator does is it codes for the RNA polymerase to fall off. And when the RNA polymerase falls off, that releases the newly transcribed RNA, which will then move away from the DNA. The RNA is complementary to the DNA, so usually they hydrogen bond with each other when the RNA is being made. And that'll happen until the RNA polymerase reaches the terminator, and then transcription stops. Any question about any of that? Here you're looking at transcription happening in a cell, and here we're looking at it under, well, that's not, this is a fluorescent microscope, where the RNA polymerase binding to the DNA and actually transcribing RNA right there. Any question about any of that? So let's look at this in a little greater detail. The RNA polymerase will bind to the promoter, and let me blow this up. When that happens, the DNA becomes single-stranded right there, and then the RNA polymerase moves down the DNA, only reading one strand, and transcribing RNA as it moves down. And it will do that, move down the DNA, until the RNA polymerase reaches the terminator, in which case the RNA polymerase falls off the DNA. That releases the RNA, which is shown right there, it's sort of hidden. And let me end this. The RNA then gets separated, and then the DNA snaps back as a double-stranded molecule. Let's see if I can get this to work. I'm going to try and open both of them just in case one of them doesn't work. Oh, wrong one. Oh, that one's not working. And that one's not working. So let me just do a search. Well, that didn't look very good. Anyone see? Oh, that's 627. That's a little long. Oh, here's the short one. And this is even shorter.
Transcription is the process of making RNA from a DNA template. Several key factors are involved in this process, including DNA, transcription factors, RNA polymerase, and ATP. Transcription begins with a strand of DNA. Important regions. The largest of these is the transcription unit. This portion of the DNA will be used to produce RNA. Upstream of the transcription unit is the Tata box. An enhancer region may also be involved. Several complexes, known as transcription factors, are required for successful transcription. The first is TF2D. A component of this factor, TBP, binds to the DNA using the Tata box to position TF2D near the transcription initiation site. Other transcription factors, including TF2A and TF2B, then attach. These complexes prepare the DNA for the successful binding of RNA polymerase. Once RNA polymerase is bound, other transcription factors complete the mature transcription complex. Now, energy must be added to the system for transcription to begin. This energy is provided by the reduction of ATP into ADP and PI. RNA polymerase then synthesizes an RNA template from the strand of DNA. Most factors are released after transcription begins. When the end of the transcription unit is reached, the RNA polymerase dissociates, and the newly formed strand of RNA is released.
Okay, any questions about any of that? So DNA, or actually RNA polymerase binds to the promoter and then moves down the gene until we reach the terminator. The RNA polymerase then falls off the gene that releases the RNA, and then the DNA snaps back. And that's RNA transcription. Any questions? If not, let's move on to protein translation. Translation is the synthesis of proteins by ribosomes using the information in messenger RNA. Messenger RNA is translated into codons. A codon is a group of 3 nucleotides. What reads the messenger RNA is actually the tRNA, and we'll talk a little bit about that. Translation of messenger RNA begins when the ribosome reaches the start codon. And you can probably memorize this one, because there's only one start codon. So the messenger, or the ribosome, starts reading the messenger RNA, and initially it just ignores the start of the messenger RNA. It doesn't do anything, the ribosome, until it reaches the start codon, which is AUG. Once it reaches AUG, it then adds the amino acid methionine. So AUG does 2 things. It tells the ribosome start protein translation here, and it tells the ribosome add the amino acid methionine. Any question about that? The ribosome then reads or moves down the messenger RNA, and it will continue adding nucleotides, or not nucleotides, amino acids, to the growing polypeptide. And when we add the last amino acid, we call it a protein. It's no longer a polypeptide, it's now a protein. And so translation will continue until the ribosome reaches one of the stop codons. And there are 3 of them, and you do not need to know any of the stop codons. Any question about any of that? So don't memorize the stop codons. When the messenger RNA is being read and translated into amino acids, it is read as a codon. And a codon is 3 nucleotides. Each messenger RNA codes for 1 amino acid. But organisms only contain about 20 different amino acids. Certainly humans and E. coli only have 20 amino acids, but there's a little over 60 possible codons. That means that there is more than 1 codon for each amino acid. Let me blow this up. There are only 2 codons which doesn't have another codon coding for the same thing, and that is AUG, codes for both methionine and start, and then there's one other, there it is, UGG, tryptophan. There's only 1 codon that codes for tryptophan. All the other amino acids have more than 1 codon that code for them. Like there are 2 codons that code for the amino acid phenylalanine. There are 4 codons that code for the amino acid serine. And then there are 6 amino codons that code for the amino acid leucine. Any question about any of that? The point is there's more than one codon for most of the amino acids. And this is known as the degeneracy of the code. Any questions about that? What happens when the DNA is UUU and the DNA gets mutated to UUC? What happens to the protein in this
No effect.
mutation?
No change.
Yeah, the protein has no change because both codons code for the same amino acid. So the protein is not changed. And we actually have a word for this type of a mutation. It's called the silent mutation, because it is a mutation in the DNA, but it doesn't change the protein, so the phenotype of the organism is not changed. And this is looking at the protein translation and a whole bunch of steps, and it's a little difficult to follow, so we'll go through it slower in future slides. Let me open this and we'll watch a little video on protein translation.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the amino acyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Okay, any questions about the video? I guess not. So let's talk about protein translation in a little greater detail. So remember, information flows from DNA to RNA to protein. The last step from RNA to protein is translation. Translation happens when the ribosomal units, there's two subunits that form together, and it'll form on the messenger RNA and then move down it, and it will ignore the start of the messenger RNA, and it won't do anything until it reaches the start codon AUG. At AUG, it will then start protein translation here, and it will add the amino acid methionine. So remember, AUG is dual function. It tells the ribosome start protein translation here, and it adds the amino acid methionine. Here's a question for you. If down here we have AUG, what's it doing? Assuming that the ribosome has started protein translation here, what is it going to do with the AUG down here?
It's going to make the protein. It'll do the first part, but not start again because it's already started.
It's already started, so it won't start here. What it'll do is it'll add the amino acid methionine. Okay? Anyways, the ribosome will form on the messenger RNA and then move down until AUG is in the, what did they call that? There's a gap in the ribosome for the tRNA to come in, and there's two gaps in the ribosome. Anyways, the tRNA will come in, and AUG has a corresponding, they call it an anticodon on the tRNA, which hydrogen bonds to the codon. So AUG will have a tRNA with UAC, and U is the corollary of A is the corollary of U, and C is the corollary of G. Any question about any of that? And as I stated, it's actually the tRNA which is reading the messenger RNA. And that reading is done by the anticodon of the tRNA. Now, one end of the tRNA binds to the messenger RNA at the codon. The other end of the tRNA binds to the amino acid that it's carrying and bringing to the ribosome. So the first tRNA brings the first amino acid. A tRNA carrying the second amino acid then approaches, and this tRNA will have the anticodon, which goes to the codon for the second codon. The second tRNA binds to the messenger RNA, bringing in the correct amino acid, which in this case is leucine. And then this methionine is removed from the tRNA and attached to leucine, the second amino acid that was brought in. And I don't really have that discussed here, so let me discuss it here. What is the enzyme removing the amino acid from tRNA and then binding it to the amino acid in the next tRNA? That enzyme is actually an rRNA enzyme. And this is the only place we will talk about an rRNA enzyme. So it's an rRNA enzyme which is removing the amino acids from the tRNA and then binding them to the amino acid in the next tRNA. When the first tRNA has its amino acid released and then bound to the second amino acid, that tRNA then leaves the ribosome, freeing up this space, and then the ribosome will move down one codon. The ribosome moves along the messenger RNA until the second tRNA is now in the first position, and then that frees up another tRNA to move in and bring the correct amino acid for the third codon. Of course, the ribosome removes this amino acid from this tRNA and then links this growing polypeptide to the amino acid in the second position here, or the newest tRNA that's come in. That frees this tRNA to leave, and that will allow the messenger RNA to move down one codon. And this process continues, building up the polymer of amino acids or polypeptides until we add on the last amino acid, in which case we now call it a protein, no longer a polypeptide. Although all proteins are polypeptides, it's just not all polypeptides are proteins. And the ribosome continues to add amino acids to the polypeptide until the ribosome reaches a stop codon. And then protein translation stops. Any question about any of that? Once the ribosome reaches the stop codon, protein translation now stops. The ribosome falls off the messenger RNA, and that releases the protein from the ribosome tRNA mixture. Any question about any of that? So the stop codon codes for the ribosome to stop protein translation, and that happens by the ribosome dissociating into two units, freeing the messenger RNA, which can then go out and bind to other ribosomes, and that also frees the protein. Any question about any of that? All right. Now, I've talked about this in simple terms to make it clear to you, but you should realize there is a complication. And that is when the ribosome is being translated into protein, there can be more than one ribosome attached to that one messenger RNA. And that means there will be several growing polypeptide chains, with the smaller ones on this end and the larger ones on this end. And as you can see, you can sort of see that in this transmission electron microscopic image. And that's just a complication of, well, actually of all protein translation. In prokaryotes, there's an even bigger complication, and that is that in prokaryotes, protein translation can start, and there might be even more than one ribosome bound to the messenger RNA. But protein translation can start in prokaryotes even before the transcription of the messenger RNA has completed. And the reason for this happening in bacteria is that this is all happening in the cytoplasm. That last part, or the last complication, doesn't happen in eukaryotes because transcription happens in the nucleus and translation happens in the cytoplasm. But in bacteria, they're all happening in the cytoplasm, and which case you can have the messenger RNA still being made, so transcription is still occurring, but before it finishes, you can have protein translation happening, and there may be more than one ribosome bound, so there's more than one protein translation happening while the messenger RNA is still being made. Any question about that complication? All right, let's talk about one more complication that's seen in eukaryotes, and that is protein translation happens in the cytoplasm, but RNA transcription happens in the nucleus. There's another complication in eukaryotes, and that is when we look at a gene of eukaryotes, not all of the gene codes for the protein or the gene product if we're talking about an RNA gene. And that is some regions of the gene we call exon, because these regions code for the final gene product. But there are other regions of the gene which we call introns that do not code for the gene product, which for most of the genes it's a protein. So an exon, that information will be translated into amino acids. The introns will not be translated into amino acids because they don't code for the protein. And when that RNA transcript is made, both the exons and introns will be transcribed, and the introns have to be spliced out and the exons spliced together. So splicing is just a complicated word which means you cut the RNA, you remove the introns, and then you splice together the exons to make the final mature messenger RNA. And in eukaryotes, only the mature messenger RNA can leave the nuclear membrane. There's a watcher or something, and these pores will not allow an RNA transcript containing an intron to leave the nucleus. And then the final mature messenger RNA will go into the cytoplasm, where it will bind with ribosomes and tRNA to be translated into proteins. And this is a complication only seen in eukaryotes, which obviously have a nucleus to regulate the mature messenger RNA to leave, and also in the nucleus the RNA transcript is modified so that the introns are removed and the exons are spliced together. Any question about any of that? You don't like this complication, do you? Because it's kind of complex. And it's almost as complex as the complexity in a bacteria, where you can get the messenger RNA while it's still being transcribed. Ribosomes can attach to it in more than one and start building the protein before the messenger RNA is made. Like I said, that doesn't happen in eukaryotes because the nucleus is where transcription occurs, and the cytoplasm in eukaryotes is where protein translation happens. All right, any question about any of these complications? If not, let me just state that we talked about the structure and function of genetic material. We talked about the flow of genetic information. We talked about DNA replication. We talked about RNA transcription. And we talked about protein translation. Our next topic, which we will talk about in our next lesson, is the regulation of bacterial gene expression. To talk about that, we'll talk about operons, and we'll talk about what operons are, and then we will talk about mutation, move on to genetic transformation and recombination, and end with genes and evolution. Any question about what we're going to talk about and what we're going to move into? If not, I'm going to end the lesson 3 minutes early because I won't have enough time to talk about the regulation of bacterial gene expression. Any questions before I log off? All right. Remember, if you have a question about the lab, to make sure you log in between 6:30 and 6:45. I'll be there to answer questions. So if there's no questions, I'm going to log off, and I'll see you guys later. All right, bye.